Olá, pessoal. Estamos de volta mais uma vez. Gostaria de agradecer a presença de todos que estão por aqui, iniciando mais essa transmissão. Este vai ser o nosso último encontro, hoje o nosso final do curso. E para fechar esse curso com chave de ouro, nós teremos então a participação do professor doutor Sina. O professor, ele é biólogo, né, e ele é formado em protistologia, ele é um protistólogo, e ele se interessa muito a respeito de ecologia de solos. Nos últimos anos, então, ele tem trabalhado com a questão do arranjo filogenético das diferentes linhagens de organismos vivos, junto com demais colegas. Então, eu gostaria de apresentar a vocês o professor Sina. Hello, Professor. Welcome.
How are you? Thank you.
Fine, thanks. And well, I presented you, as I said before, the basic stat, but if you want, you can complement your formation and you can start your presentation when you want, okay? I will be here in the backstage and thank you for your availability.
Thank you so much. And thanks for inviting me to do this. I can't see everybody, so hello, everybody out there. I'm in Canada right now. I'm in Saskatchewan, and it's the beginning of winter. So today is 5 degrees, and at night it's usually about -5, -10 at the moment. And it's morning here. So I'm going to give three presentations, and the last one is a bit longer. First, I've provided a bit of a historical perspective on how we started to work on modernizing the classification system. And then I will talk a little bit more about some of the lessons that we learned and how we're planning to go forward. And last, I will give some examples of the sort of ecology we can do working with produce communities. So first, I'll begin with this presentation, which is a bit of a overview starting back many years about what the problems were and how we started to learn how to move forward. So this was the 2012, so the second revision. The 2005 was the first one. And this is a figure that became popular and many people used. Gives you an overview of how things were understood around 2011, 2012 when this was done. And this is roughly what the situation was in 1998, which is when I finished my PhD in Vancouver at UBC. We had a classification system that was published in 1980, and this predates all of the molecular biology and phylogenetic trees. So it was the last of the old system, and it was difficult to modernize it because the phylogenetic trees at the beginning in the late 80s and through the 90s, we were still trying to understand how to understand the phylogenetic trees. And so there were a lot of arguments about what was the right tree, especially at the base of the tree. But around 1997, 1998, at several symposia, we started to realize that the phylogenetic trees from the different laboratories were increasingly the same, and we were starting to understand how this was going to assemble. And the first one is 1997 by Yves van de Peer, where they connected many computers together and did a very large calculation and came up with a tree, the largest tree that was possible at the time, where the map of relationships between protists and all the eukaryotes was assembled, and this was fairly accurate and showed people what we could do going forward, especially as computers started to improve in their memory and speed. So this was the first complete phylogenetic tree of eukaryotes, and it was surprisingly good. Even when we look back now at this older paper, it was quite a good effort. But ISOP, which is our international society, had not been very involved in this, and the journal Eukaryotic Microbiology hadn't been publishing many of the important trees because they were being published elsewhere, and the discussions were happening at ICEP, not at ISOP. And most of the papers ended up in IJSEM. So it was a period where there was a lot of arguments between laboratories, and one of the most troubling aspect of that is that we lost name stability because the families and orders were being moved from one phylum to another and one part of the classification to another part. Everybody was inventing new names every time something was moved, and different laboratories were using different names to describe the same thing. And when at conferences, for the older professors, it was simpler to say, well, in this lab, when they say this word, they mean these things. In this other lab, they use this other word, but they mean the same thing. But for a new graduate student, it was impossible to understand their presentations because you didn't really know what they were referring to when they were using different names. And so without having a protist textbook and without having a reference classification system and a standardized set of names, at the end of the 90s, early 2000s, communication at conferences was very hard because different labs were using different vocabulary to describe the protists groups. And so this eventually started with Mark Farmer and myself, who were both at the University of Georgia at the time. He was a new professor and I was a postdoc. And we started to make drawings at lunchtime about what the protist classification would look like. And we did this for a couple of years over coffee and lunch here and there. And then I went to become an assistant professor and he became department head and became more busy with administration. But we continued to work on this nonetheless and tried to work towards the publication of a modern version of a protist classification. And some of the hurdles were that the ranks were a problem. Nobody would agree on what rank should be assigned at nodes on trees. There was also no agreement on names because different laboratories had introduced different names for taxa at various rank levels, and they all wanted their name used, but obviously you have to choose one. You cannot have 5 names. And the worst part of it is that groups that were being treated by the zoological code were being moved into groups where the botanical code was being used, and these became very intermingled. And it meant that you could, even though having 2 codes for protists was a problem before, it became really, really difficult, if not impossible, trying to assemble a new classification with this system. And so this led to about 5 years of a lot of emails and correspondence and discussions at conferences. And one of the reasons why many people were hesitating to get started with it was because the illustrated guide 2nd edition, which some of the famous people at the time, like Cavalier Smith and Patterson and David Lynn, Dennis Lynn, were engaged in, could not come to an agreement on any names and structure and format for the classification. And they never spoke to each other again after 1999. And there was a lot of very bitter arguments as a result of this. And so a lot of people were very shy about venturing into this field. And so working quietly with emails and behind the scene, I think we managed to work around people who had too much ego and would not compromise with anybody. And we began working only with phylogenetic trees that were robust and working through hypothesis testing and slowly assembling classifications, and then secondarily looking for the oldest name that correctly groups the genera into clades. So this was very systematic, and it set aside who invented what name in order to really focus on what the phylogenetic trees were telling us. But one of the obvious things is that you cannot use the zoological or botanical code because they are incompatible, and you have to bring back name stability. If every time you change the rank of something, you change its name and authority, after a while, nobody can keep track of which group of genera we're trying to talk about. And so this makes things difficult. It's easy over 5 years, 10 years, by going 200 years back, trying to figure out what was called what. Is difficult if you don't have access to the original material. And increasingly, many of the type specimens were not very useful. One, because people were damaging them trying to extract DNA, and two, they weren't showing the correct morphology sometimes, and then trying to find the type specimen in the original locality was sometimes not possible because the streams or villages no longer existed. And so these problems with type specimen and changing of names every time you change ranks really make things difficult. And so as we move forward, one of the lessons was that with some people, you just cannot work because they're disruptive and they will not listen to other people and other arguments, and they just want to do things their own way. And so some egos were really in the way of moving forward. So we didn't work with them. And then during this time period, things started to come together. Once we set aside these problem individuals, things moved along much faster. And we were left with the idea of which rules should we be using if we cannot use the botanical code or zoological code. If we pick one, all of the botanists and phycologists will get really upset. And if we pick the other, all the zoologists would get very upset, because these have become cultural. It's how you learn to discuss things and how you've memorized things. But one of the emerging problems was bioinformaticians, because these were individuals with very little biology background at the time, who were good bioinformaticians, but they didn't have any understanding of what these sequences and genera names were. And sometimes things were mixed together erroneously, and bioinformaticians couldn't see that biologically it makes no sense to mix these groups with these groups. There's a statistical error. And so many papers were being published by bioinformaticians who didn't understand the biology and evolution and why that was impossible. So those kinds of papers you also had to remove from your analysis as you move forward. So here I'm going to introduce some terms, and these are the codes of nomenclature which are kind of relevant. There's the bacteriology one. There's the phylocode, which was early stage development. It was still an idea in those days. And these were the ones that were in effect. The phylo-code was really a very early stage development, and there was a group of us working on phylo-code in the background and having meetings every so many years to try to move this forward. Here, some more definitions. Systematics means according to a system. Doesn't have to be a botanical system, or it just has to be a system. So you need a set of rules that you adhere to. Taxonomy is a system for classifying. There are rules about how to name things. And classification has to do with how to arrange these into categories. So traditionally, species are put into genera, and genera into families and orders and so on. So that's kind of, and the shift that was happening in Linnean time, without the concept of evolution, things were grouped together based on whether they were morphologically similar. But increasingly, especially by 2000, people were expecting the classification to reflect the phylogenetic trees, which reflected evolution by common descent. And so there's a shift here in perspective that happens late in the 20th century that was very different from how it was being used in the 19th century. And so nomenclature is a system of rules for naming these things. And so we have societies of nomenclature, we have systematics and nomenclature societies that deal with these things. Some of the problems in trying to use the botanical or botanical zoological code is that Linnaeus was basing his structure on Aristotelian understanding of the world, and these predated the idea of common descent that was introduced by Darwin in 1859. Linnaeus and Aristotle still believed that there was a fixed number of species, and then we just have to arrange them into lumps. One of the problems of the traditional approach is that every time you move something, if you move a family to somewhere else, or if you change the rank of a family, then you also have to change the ending and the name. And this causes a cascade of name changes elsewhere in the classification. And in this traditional system, there's a lot of emphasis placed on the authority, so that you always have to have a name followed by the authority, and less emphasis placed on name stability. And this was also a handicap. As you'll see in the modern way of doing this, we kind of pay less attention to who changed the rank or the name, and we focus on trying to retain the name stability, even though when we move something through in the trees. There are other issues that prevent these from accommodating each other. One of them is that ICBN still insisted on having everything described in Latin, but the zoological code no longer did. And I was part of the committee on algae for the botanical code. And I was there when the proposal to abandon Latin failed. And then at the next meeting, about 4 or 5 years later, that motion passed, and ICBN finally abandoned using the Latin. The other important issue that we still haven't addressed properly, and I think UniUc and UCREF are probably going to help in moving this forward, is about type specimens. The rules for what consists of a type specimen are almost useless in the modern world, because they will typically be cultures or slides. And if you're working with an amoeba, with a slide that was made in the 1800s describing a new species, you just don't have enough morphology with a little bit of cytoplasm that is shapeless, and a nucleus, and hardly any other organelles to work with, so they all look the same. So even though the type specimen exists, it provides no useful morphology and no accessible DNA. And so a lot of these type specimens are not useful. And if you're dealing with something that's mislabeled, then there's absolutely no way of sorting your way through it. And so the codes failed to provide name stability through the centuries, and this is something that needed to be addressed. Modern biology relies on electron microscopy and DNA sequence information, and this is where type specimen information should be focused. And whatever classification we have, the system behind it, the systematics, has to accommodate changes in position and rank without changing the name. And that's easier if you're not changing families and orders from one phylum to another all the time. But because of the amount of change that happened in about 20 years with the molecular phylogeny information, there was just too much, and the codes could not handle that amount of change. And so you have to come up with a new set of rules. Because the loss of species name, when the species is moved to another genus or the genus to another family, is a very bad system for finding files and finding names again. And you have to rely on people's memory, but when you go back 200, 300 years, it becomes a real mess. So one lesson here, the third one, is that naming clades correctly has to be a community effort. One person naming something and publishing it and expecting that everybody will use it is naive. It's misguided. Sometimes people will disagree with it. Sometimes they will disagree with the reasoning behind it. And it will be another name that becomes dumped. And the correct way to do this is to propose names informally and have them discussed with the community and come to an agreement and an understanding about what might be a correct way of naming things and a correct way of moving things. Otherwise, you end up with a lot of errors that need to be corrected, and this is time consuming and generates a lot of unnecessary corrections. So building a new system, some of the things that needed to be stable were a way of having formal names and designating group names and authority with definitions that were clear and definitions that work in the modern world. So not just type specimens, but you need to have some electron microscopy, some photos, some maybe video footage if it's an amoeba, DNA sequence information, and some discussion about which DNA sequence part of the DNA would be useful. Not all of these were agreed upon in the early 2000s. Clades, they have to be monophyletic, and that by definition a clade is monophyletic. But before the mid-2000s, many scientists did not have a problem lumping things together that were not monophyletic, and that was not acceptable for a lot of other biologists who preferred to be strict cladists. These debates, I think, are forgotten, but they caused a lot of anger sometimes at conferences between people. And so the system that emerged out of these 5, 6, 7 years of discussions was one where the ranks are nameless. There is no ending to specify a rank. If a name has an ending that specifies a rank, you ignore it. It's just an artifact of history that ending is there. What you're really doing is focusing on what genera are grouped together into this category, and you give it a definition and you give it a name. And if you move this box that has these genera in it somewhere else in the classification, you don't change the name of the box and you don't change the name of the genera inside it. It remains the same. module that you move in the classification system. And this is very important for preserving name and on remembering what genera are in this box. Even if you change genera, the name of the box doesn't change. So this was a very important fact to accept, an important rule to accept moving forward to return to stability. And when you choose people that can work together, you can make this sort of progress, but you have to understand that there will be people that will refuse to work cooperatively and people who are not interested in other people's opinions. And that doesn't allow progress. There were a variety of proposals published about what to do when a species is moved to a new genus. So if I have Paramecium aurelia and I move Paramecium aurelia to be, for example, in Tetrahymena, then it becomes Tetrahymena aurelia, and nobody will remember that old papers that say Paramecium aurelia is actually today Tetrahymena aurelia. This kind of changes means that you lose the thread of history unless somebody remembers and somebody goes and finds the papers. This is harder to do the further back in history you go. And so there were suggestions that if that this might not work very well, that if we move this pair together or this pair together to a new or this way, then we have the new genus here and we keep these paired words together so that we don't lose track of things. There hasn't been any agreement on this because it hasn't really been used, but this is one of the last items that we're going to have to address. The other thing that was interesting is that a lot of people were beginning to accept the idea of nested clades, which is what Phylocode was trying to develop, and the system that Phylocode did in the end develop. And the advantage here is that you separate the naming of the clade from the assembling of the hierarchy. So this is what I was talking about, where you have a box that has a name with some species and genera in it. And when you move the box, the name doesn't change. So as long as there's no hierarchy linked to the name, you can do this very easily. And this is something that I think most modern biologists have now accepted and do it this way. So I think as lesson 5, I've written that one of the important lessons really in the mid-2000s was that people who do not have an open mind and are not amenable to change their thinking are not really scientists, because we do science in order to progress, in order to understand things better. And the only way you can have progress if you are willing to do things differently. Just because you memorize it one way doesn't mean that it was the right way, and the way we see the world will change after 10 years, 20 years, 50 years, and you have to be prepared to relearn very fundamental things as we understand more how things work. And so identifying people who do not have an open mind and are not willing to accept that there is progress, technological change or conceptual advance, makes it very difficult to move forward. And so you have to identify people who are not very good scientists because they're always blocking progress. Another thing that emerges is that more and more we have people from many different countries and backgrounds doing phylogeny. It's taxonomy and biology and classification is no longer a European thing the way it was 300 years ago and 400 years ago. And we have many people from Eastern Asia who are biologists, a lot of money being spent on research. And when we name things and describe things, we have to understand that it has to be phonetically pronounceable with ease across cultures. It has to be translatable, and some names are not, and so they're not very good names to choose for your clade, the taxa. And for type specimen, this is something that I think there is still some discussion, but there's progress, and things that I keep pushing is that we need more and more to rely less on morphology, but on biologically useful information, on choosing the correct DNA sequence for the group. And we need to have digital images. A lot of these old microscope slides from 200-300 years ago are not very useful anymore. And if you go to museums where there are specimens collected of algae, those are very valuable, but it's much harder to do when you're dealing with a 5-micron little flagellate or amoeba. And then the other thing is that it must be public and free and web-based, so that everybody can access it. You cannot have a private organization have all of the type specimens and then you have to pay for access all the time. That just will not work. And so whatever system we develop for curating this has to be free, easily accessible by the web. And that's what the UniUC and UCREF are trying to accomplish. So I think I'm going to finish up here with this kind of historical overview. And we, in the 2005 paper, we came up with a name list with ranks that didn't have any endings, but had formal definitions. If there were polyphyletic groups where the monophyly was still in doubt, those were indicated that they are not monophyletic and probably will change. And we made a very hard rule that the oldest correct name has to be used, amended if necessary. And then there's this thing about empty ranks, which I will talk about a little bit more in a few minutes in my next presentation. Empty ranks is when you have a genus, and it's a single genus in a single order, in a single class, in a single phylum. And some people like to call these things kingdoms. These are empty ranks because in reality, you just have one genus. It's not a kingdom. And so if you use strict botanical zoological code, many people will insist on putting these empty ranks in, developing new names for phyla and classes for just one species or one genus. And this is viewed by the community in general as a very, very bad idea. And it's people wanting to name kingdoms rather than people helping progress and classification. And as I said before, the endings in ranks have to be considered an accident of history. It's just part of what was at one time useful in the name, but is no longer relevant to indicating the rank, because that will change as we describe more genera, and there are a lot of unknown diversity in the world. And as we work on this more, there will be more and more changes to our two nodes. And so we have to accept that as we are going to be moving and modifying the phylogenetic tree and therefore the classification, we have to do this without changing names all the time. These issues were discussed in 2007, right after the 2005 paper. And I want to bring to your attention here the phylocode, which I'm going to touch on again. The phylocode was in development through the 2000s and also through the 2010 to 2020 period. And we had meetings here and there, and even though this was a very small group of biologists, about 50, there's about 20 years of work that has gone into publishing this, and it's now published. Many papers, especially by Cantino and De Curas, discussing the phyllocode as it was evolving. And this is a code of nomenclature to replace the botanical and the zoological codes to deal with modern techniques and focus on apomorphy, branch-based, node-based definitions, and groups are related by common descent. You really have to work on the phylogenetic tree and then use that to develop a scientific classification. It corrects most of the problems with the codes. There are problems that remain with what is a species and how to define and name species. That I think is something that, as I've said before, we're still having to discuss among ourselves. This is where the phyllocode runs out of steam. De Curas and Cantino have now retired, and so it's kind of us as the next generation that have to continue this work. So the code, when I prepared this, I said is ready but not implemented. But in fact, last year the code became implemented. So now we have the phyllocode. This I'm going to skip over and point out a little bit what the difference is between a phylogeny and a classification, in that the phylogeny guides the classification. Many people think that they're the same thing, but they're not. The phylogeny is looking at the wrong end of the tree. It's better for putting classes into phyla and phyla into larger groups. But when you're looking at species and genera and families, you can only work with a small group of them, and you have to, and it becomes more difficult to have good species representation across all eukaryotes if what you're really interested in is the details in one family. And so here, a lot of the common terms and informal names that have evolved in laboratories have to be replaced with formal names. And what phyllocode provides are definitions that if suddenly the group no longer meets the definition, then you no longer have a monophyletic group. It's not a clade, and you have to come up with a new definition for it and put it in its correct place and position and with genera composition. So that's kind of where this ends, is what are species? And that's roughly where something that we're still dealing with, but I think we're in a much better position now in the next 10 years than we were in 2000 or 2010. Most of that is technological developments and what we can do with bioinformatics and what computers can do. We have a better understanding of which DNA sequence and how much sequence divergence. It's not the same across all the groups. In some amoebae, for example, a single nucleotide change is enough that you can be certain you're not dealing with the same genus, but probably the same family. In other groups, you can have 1, 2, 3 nucleotide change in a particular DNA sequence, and that is considered normal differences between populations and closely related species, and doesn't make a new genus. And so you have to have some understanding of how much variation and the rate of evolution is in your little group in order to be able to make this kind of interpretation. Other issues that we're beginning to grapple with is about biogeographical sampling. What is endemic? Which genera or species can be found everywhere or almost everywhere? And I think young biologists today are a lot more aware of the fact that just because something looks the same at the microscope, it's not going to be the same species. It very probably is, it could a cluster of species that look identical. And we have many, many examples of this. this was much harder for a lot of the more senior biologists to accept in 2000, but I think today almost everybody understands this. And evolution through selection acts on ecological parameters. Biologists that do molecular phylogenies are not very good at collecting ecological parameters when they're sampling that will become useful in understanding species function in a food web and their biogeography. So these are the sort of things we have to become better at doing. And so last lesson on this topic is that as we're progressing, the field is shifting in how it understands biogeography, what a species is, and how useful the species concept is when we're dealing with organisms that are very small and have very little morphology to go by, and how much ecology you need to understand in order to understand what different species are within a genus. So I'm going to have a pause here. And if you have any questions about some of the things I've said, this is a good time to ask them. We can take a 5-minute break. You can ask some questions, and I want to pick up on some of these topics and talk about them a little bit more in the next set of slides, which will take about 20 minutes. So if you have any questions, please bring them to me. I'll be happy to answer if I can.
Ok. Pessoal, vocês têm alguma pergunta para ele? Nesses 5 minutinhos. Dr. Sina, well, while we have not the complete conditions to know exactly what are the limits of a species, what do you think that is better, split or group species?
I think it partly depends on which group of organisms you're working with. In what we're beginning to see, I think, from biogeography and global sampling efforts is that we have genera that are marine, we have genera that are terrestrial in the soil, we have genera that are freshwater. And in some genera, there might be species that you can find in the marine environment and in freshwater or marine environment and soil, but those are rarer. Most of the time, the species in one genus are in the same environment. And sometimes all of the genera in one family belong to an environment with no species in another environment like a soil or freshwater. And so I think the amount of variation there is between species is also very different. If we look at sexual groups in ciliates, for example, there are about 30 identical tetrahymena species, but their species can be differentiated not by morphology or DNA sequence, but only on mating clusters. And the same is true for another group of ciliates, the Euplotes, have these mating clusters where some are compatible with each other and some of them are not. And you have populations that mate with some populations, but not with other ones. And so it becomes very difficult to know where the species ends. What we know is that we have a good genus, but where is the limits between the species when there is no clear-cut mating cluster? And these we encounter in mycology. We encounter it across the protist classification. And so I think it really depends on what you're looking at, which group is that you're looking at. Sometimes it's very clear cut that this species is these populations, and you find them here. And if you look at the ones over here, they're quite different in ecology, if not also in morphology and ecological functions. So I think it really, because the protists are so diverse, I don't think there is a single way of doing it.
And we have a complementary question.
Mm-hmm. Another question?
Because it's here in the screen, but I will read for you. Controversial question on species concepts in protists, or for everyone, real or artificial? Does it matter if it's one or another? Thanks and great talk, Professor. It's a messenger from Felipe Benitez.
Thanks. I'm not certain if I quite understood correctly, but I will try to answer. I think the delineation between species is probably not that important. If we have an understanding of how much variation there is between different sample populations, what we probably should be focused on is the ecological function. What does it eat? Under what conditions is it active? What are the preferred months? What are the preferred depths? Does it need more or less oxygen? Those kind of ecologically useful parameters are probably more important in understanding how the community and the ecosystem work. And trying to decide if there are good species and where the species delineations are is probably a lot of work that is not very useful.
Great. Oh, we can continue?
Sure.
Okay.
So I'm going to pick up on some of these themes.
Okay.
Yeah.
Perfect.
So I'm going to go over this. This one is shorter, but it kind of puts us a little bit more in the technical way of approaching it, and how we are trying to address that with the philo code. So if I take you through this, can everybody see this okay?
Yes. Yes, the first one. Okay.
So we're looking at the diversity again, and sampling, and trying to decide when you have new species, or if you have samples, you have a DNA sequence, you identify it to the nearest genus or species in the database, and you have to work with that. There are different ways of doing it. So if we look at the assembly of a phylogenetic tree, and then from phylogenetic tree to classification, what I have placed here are some species that are grouped together like this into genera, and we can put these genera together into families. And it doesn't matter what you call these things. If you don't like ranks, these are your OTUs, or your species, approximately. And then this level, I think almost everybody will agree, is called a genus. And then these become more problematic. Some people will want to call them families, or maybe they will say these are subfamilies inside this family. Someone else will say, no, these are two families and this is one order. Someone else might decide this is a suborder and this is the order. And this is why the higher up you go in the tree, the more debate there is on the rank. It has to know with your experience with these species and how you understand how they should be clustered. And this is a very real everyday problem for biologists, for protistologists. So here you have your species, and then there are some ranks. There are nodes on a tree. Some people think that every node must have a rank. Some people will say that only some nodes need a rank. The other ones are useful. We know that these cluster together this way and that way. But for a classification system, you don't need to name all the ranks, only the ones that are useful for teaching or curating a museum. And these are real arguments. I don't think we can say this is correct and this argument is incorrect. I think these are real everyday decisions that we have to make and work with. So the simplest system from a Linnean perspective is one where you have species that are clustered into genus, family, order, class, phylum, and then later people started putting these into kingdom and things. But this is very simple, and it worked well a few hundred years ago. Today, it's a little bit more complicated, and the complexity really comes out of trying to understand evolution and common descent, and trying to have a phylogenetic tree that represents the history of the evolution, and then translate that into a useful classification system. So what I've pointed out to you is the kind of arguments people get into about whether are these subfamilies and is this the family, or are these two families and this is an order or a suborder? What do you call these? This is where people like Cavalier-Smith and a few others were always arguing with other people because they wanted it to be this way and other people thought it should be another way. And to most people, this is arbitrary. And it's not just arbitrary, it's almost irrelevant whether you call it this or a suborder or a subfamily. What matters more is, is there a real node or is there not a real node? What's the robustness of these nodes? That's the biologically useful statistic, and not whether it's a family or a subfamily or an order or suborder. Those are kind of almost irrelevant. And it gets more complicated. I've got one example here with a ciliate and a species. So Amphoreilopsis tintinus belong to Tintinidae. And then you need all of these ranks to arrive at what everybody will accept is a phylum. So what do you call these? Do you start with genus, family, order, class, and then have all kinds of superclass and infra-kingdom and parv-kingdom and whatever to arrive? You have to come up with names to fill this. And some people will argue that, well, we just put more subfamilies and suborders and try to reach up there. And some people say, well, we just call them subphyla and subclasses and suborders and work our way down. There's not even an agreement on whether you should start at the bottom or at the top. So this is a paper that started a lot of the disagreements and problems, in that when this was put together, it was done by one person without discussion. It was supposed to be a committee effort. It wasn't. And this kind of interpretation in 1993 predates when we had appropriate bioinformatic techniques. Primitive computers, very little taxon sampling, very poor understanding of what a change in a DNA sequence meant, very poor understanding of which DNA sequence was relevant. At this time, a lot of people were trying to use alpha tubulin or beta tubulin or some metabolic enzyme as the best way to understand molecular phylogenies and to build trees. There was a lot of discussion about whether it should be the small subunit or the large subunit of the ribosome as a better tool, and which part of those ribosomal DNA sequences should be used. Those have been in discussion since we have made a lot of progress, but you have to see under what technological limitations and information limitations these papers were being assembled at the time. This is the system that was used in that paper with empire, kingdom, subkingdom, all these terms introduced to try to have enough nodes to deal with species. And you can add more of these intermediary terms here. One of the problems this does is in trying to extend the Linnean codes to molecular phylogenies is that we have a lot of nodes, and we're not always sure which one is a solid node until some years later. And every time you have to make a change, for example, an order becoming a subclass or an infraphylum becoming a superorder, you have to rename everything. And then you lose track of what was what if you make these changes 3, 4, 5, 6 times over 15 years. Who remembers what the sequence was, especially when you're a new graduate student? You know, I can't remember up to a certain point what people were publishing in the 90s, but I cannot do it for what was being published in the 1860s very easily. It'll take me months at the library. And so here we have 20 terms introduced, similar to what I showed you here, where you need a lot of names for all these different nodes. And some people started to say, well, we don't need to name those things. We can work just fine without them. This is the first paper, and this is a historical paper you should probably know. It's the first one that looked at eukaryotes, and they were using 28S ribosomal RNA. So it's André Adout's lab. I did a postdoc with Baron and Perrasso, and Adout passed away just about that time period when I went into the lab. But it was too much too soon. Here in this paper, the taxon sampling was very few. They were walking on the beach and picking up stuff at random. They went for a walk in the woods and picked up stuff at random. And then they were using bioinformatics. There were some misidentified cultures. There were contaminated cultures. And the misidentification and contamination plagued a lot of the work in the 90s and early 2000s, where people thought there was one bug in the test tube, and then suddenly you realize that, well, there are a bunch of bacteria problems, there are cysts of other things, and sometimes there was another little algae that you couldn't see and didn't see because it was stuck on the glass. And all of these problems plagued the 90s and early 2000s. So when you read those older papers, you have to remember that there were a lot of problems to resolve. Don't just read a paper and say, this is what they said. Look at who cited the paper and whether there were problems with the paper, because often there was. And so another lesson here is that speculation belongs in papers, in the discussion. You don't translate that into the classification. Classification has to be a higher bar and much more robust and much less dynamic. The purposes are different. Phylogenetic trees you build to make progress in understanding evolution. Classification is for classifying. It's a way for you to remember stuff in your head. It's a way for you to categorize stuff when you're at the museum. It's a way for you to find things in the library and to retrieve things in your head. It's not the same thing or has the same purpose as a phylogenetic tree. These are different things. And so more and more, I think reviewers were a lot more lenient 20 years ago, 30 years ago, in letting people come up with not very good phylogenetic trees and making change in the same paper with the classification, and then use these errors in subsequent papers. I think now reviewers are a lot more strict about this, and you can discuss your phylogenetic trees as much as you want, but to make changes to the classification is not something one person can do in the discussion of a paper. There has to be community discussion, and sometimes 2, 3, 4 years of it before you have enough people agreeing that, yes, okay, we can revise this part of the classification. Otherwise, you're dealing with things that are not robust, uncertain, speculation, and you invent a lot of new words for no reason. So it has to be discussions with the community at conferences. That's why you have beers with people. And this is what I talked about before, that there are different cultures, there are different languages. For example, the botanical code doesn't speak the same language as the zoological code. And if there are different ways that these different cultures do things, the way a mycologist will name things, especially at the genus, species, and population level, is different from the way a bacteriologist will approach it. And the other thing you learn when you work with people who have different cultural backgrounds is that they will get very, very angry if you tell them that their botanical code names are going to be changed to zoological code names. It makes people very upset. That's why it's cultural. And it is part of how you were brought up. It's how you were taught. You belong to a community that gets together at conferences, and you talk about things in a certain way, and you have a way of practicing that discipline. And when someone comes along and says, well, we're going to use a different system with different vocabulary now, it's always upsetting to everybody. And the other thing is that you're changing the story, you're changing the origin, you're changing how things are going to be done, and you tell people, well, the way we're doing it before is wrong. We have to do it this way. Whether you're dealing with politics or classification, people behave in the same way. And these have to be done slowly over time, with an entire community coming to grips with the fact that there is progress and things have to be done differently. It's not something that you will fix in the discussion of one paper. So this is a theme that I talked about before, and I call it the Linnean curse, is that every time you move a clade, you change the name and the authority. And over 100, 200, 300 years, it becomes very difficult to trace back who meant what by a particular species or genus or family. So here's an example here. You move it over here and it becomes Stokesia aurelia. And then you move it this way with the yellow. So phylogenetic trees change the tree here with more taxon sampling and better understanding of the DNA sequence information. And then what you have is two changes, and it becomes harder to track. So if you have a student looking at this system and another person comes and says, well, this is the same as Paramecium aurelia, this student will not know to go and look for Paramecium aurelia papers if they're interested in working in Stokesia aurelia. After 10 years or 100 years, this memory becomes lost. And all of the classification is full of this. And that's why name changes and stability at naming the genus and epithet is very important. And there has to be some agreement on how we're going to do it. So it's a bit like changing the words for stuff. You know, if you're used to calling a chair and somebody comes along and says, well, from here on we call it a table, it's very disruptive to the language. But that's what we're doing to the classification all the time. And then when you change the rules, you're changing the grammar. These are the reasons why when we're dealing with classification, there's a lot of culture involved. There's a way we speak about things, there are words we use, and to have botanists and zoologists accept that we cannot have two ways of talking about the same thing in the same country or in the same government, it is a difficult thing. I think we're not where we were in 1980, where everybody was fighting with everybody and throwing things at each other at conferences. It's not like this anymore. But we still have some serious talking to do about how to retain name stability and which rules to use when we call something by a nucleate name. So the point I'm making here is you can't change language every time you change rooms. You have to have a way of communicating over time. This is a fun paper. If you want to read it, 2006. It's a fun paper because it raises a very important point, and it raises exactly the kind of issues that I just talked about with the zoological code and botanical code. Here we have a Pneumocystis, has always been studied and taught by protozoologists with a zoological code. Molecular phylogenies place Pneumocystis without any debate within the fungi. So suddenly the mycologists have to look at it as belonging to them, and the botanical code applies. And I've given you some examples. The reason this becomes important is not because we're changing Paramecium aurelia to something else. There are a lot of parasites, human parasites and some animal parasites have a lot of legislations from governments about how they should be treated. If you change the name, you have to go back to the parliament and change the legislation. If something is treated as a protozoan parasite by your government, and suddenly it is a fungal parasite, it has to be treated by a different set of rules, and everything falls apart because the agencies that are monitoring this disease are no longer responsible for it, but another agency that before never had parasites suddenly has parasites that it has to work with. These changes for parasites have cascading effects all the way through administration. It's a real headache. You have to rename and rethink all of the type specimen. And so these repercussions through the system, when you're dealing with species that are legislated, is a big deal. And it's not just with parasite, it has to do with endangered species. There's legislation around conservation of endangered species, including international legislation. You start changing these names and you say, well, this and this red wolf is actually a hybrid of a coyote and a wolf. It's not a new species of wolf. Some people will say, well, it's a subspecies of this or a subspecies of that. From a politician and legislation perspective, this is very awkward. They don't know what to do with it. They don't know if they should protect hybrid species or if they should not protect hybrid species. And this is where the language about what we call species and subspecies and populations takes on a different proportion. It's not just about a different strain of a parasite anymore. It's about how many millions of dollars you're going to give some organization or agency to monitor a particular thing. And if you take the money away, they're out of a job and there's no species to monitor anymore. And it's the same when you're dealing with toxic algal blooms and endangered fish and endangered coastlines and things like this. These can have repercussions, but they're day-to-day nitty-gritty stuff that biologists have to work with all the time. So they can have consequences. These I've talked about. I don't want to spend more time on it. But I want to remind you why the ranks were a problem and how we ended up with not using ranks. And this is how we ended up having to use those darn bullets. Those classification papers have these bullets as a way of indicating nodes and change of ranks. And it was a way of to appease everybody, where you recognize rank and nodes, but without naming them, and you can still find your way around the thing. But it becomes very hard because when you have 1, 2, 3, 4 is one thing. When you have 7, 8, 9 bullets, and you have to start counting those things, it's just terrible. This is not the best way to do things. But it's a compromise, and it has been picked up by a lot of people. This is the 2-volume edited book that was published now 3 years ago, by Archibald and Simpson and Slamowitz. And they used the same system again, because the book authors, none of them could agree on what was phylum and class and order, but they could agree on the name and the ranks. And so this system becomes translated and used by books in order to deal with this problem. And so on a day-to-day basis, what you have is how do you assemble a tree? And I've talked to you about people wanting to start at the bottom with species and genera and group them on the way up. And some people prefer to do it this way. And neither way do you have enough ranks to reach the top. And so you come up with intermediary names. And then you have this other problem that I talked about, where you have a species. It doesn't fit anywhere in the classification. Something like Malawi monads, for example. And then you have all of these ranks that have nothing in them besides one species at the bottom, and you have to come up with new names for every one of these things. And then one day someone says, oh, well, Malawi monas, if you look at it this way and do the bioinformatics this way, it actually belongs over here very neatly, and we don't need all these names. And that's something that infuriates a lot of people, because if you have a problem like this, just leave it like that as a species or a strain, an OTU that you cannot place somewhere. Eventually you will find the home for it. You don't need to do all of this and cause trouble. Eventually we'll put it where it belongs. It just takes a bit more work. So these monotypic kingdoms, for example, are usually treated as inserted cities by people who are a lot more reasonable and rational. And then you get people who argue about my 5 genera belongs to one family. It's the only family, but it's an important family, and so I will call this family a phylum. You see these kind of arguments. These are often people trying to get the attention of reviewers and grant proposals and making what their work sound more important. But from a publication perspective, I think all of your colleagues will probably say nasty things about you when you're not there, when we behave this way. So I've said this before, and I'm saying it again, a classification is not a phylogenetic tree. They have different purposes. They're different things. This is research, and it leads to progress. And this is for teaching and curation and memory. They have different purposes. And not all nodes need to be transferred to a rank in the classification. You just need enough to be able to remember how far down in the classification you are. So these are things I don't want to spend too much time on, but I'll put them here just for you to know where they are. There are some misused terms in protistology. and they were defined. The locomotory ones were in the 2012 revision. The 2019 really focused on functional groups, on what is mixotrophy, what is cytotrophy, and all of the more ecological terms that are required. But the other thing that the 2009 revision included, which have turned out to be useful, is to put number of genera in the clades, so that we have a better understanding now of which groups have very few genera, which groups have many genera and a lot of diversity. All of them were assigned functional groups so that we can do ecology and write better interpretations of our community ecology discussions. And we also introduced all of those terms in the classification into Chinese script. The Chinese script, so used by Japanese, Korean, Chinese, and countries, were still using terms from 200-300 years ago, and they needed to be adapted to the modern protistology vocabulary and understanding of the trees and classification. So we introduced a lot of new terms and corrected the writing in Chinese script of a lot of these terms, and this has been remarkably useful. I thought I was going to get a lot of hate mail from this, but it seems that when we did this, it helped them move forward, because they didn't have to argue among themselves how to do it, and a larger group of international people did it for them, and it has settled everything so that now they can use these without having gotten into a discussion and disagreements amongst each other. So these were good things that came out of it. And so I want to finish this part by talking about work that Colombo de Vargas led with the support of the Moore Foundation. And this is this curated protist database system. Many of you have probably had to work with this. It has various parts, and it took several years to get this developed to a point where there was sufficient funding, and then to work on the structure of the database and to link these different parts together. So this part here was a result of several meetings, face-to-face and conferences, small group of handpicked people. The UCREF is one thing that came out of it. The UC Bank, which is the curated DNA sequences, comes out of GenBank, and this has been this was done also at meetings where we had experts from each little group there curate what was in GenBank in this new data bank. And UCMap, which is will take some time to evolve and produce, is trying to link photographs to OTUs and more metadata. So these are things that we're going to see mature over the next 10 years. Lastly, the phylocode, which is now published, and it's an additional code of nomenclature that can be used. More and more people are switching to phylocode because it avoids all of the problems of the botanical code and the zoological code. And the way the 2005, 12, and 19 revisions of the classification were done were increasingly worded in phylocode-friendly vocabulary, so that the transition to phylocode is simple. Alistair Simpson did a lot of the translation of the definitions for taxa and clades in the book, which is the phylonyms. And so this is probably something you should become familiar with as we move forward, because it will be used more and more. The regnum here is where you go to enter your new name and register it in phylo so that it becomes formalized when you enter it, and it's curated. So you can't just go in and type it in. Someone will look at it to make sure it has all of the correct information. And the way most biologists work today, and these are recommendations from phylo is that you have to have a phylogenetic hypothesis. Doing it the way Cavalier-Smith was doing it will no longer get you published. If you have results that are correct and the node is robust, then you need a node-based definition. If the hypothesis is defeated, then this evolutionary scenario was false. The name for the failed group disappears. You might be able to use it in another context, but it gives us scientific hard ways of, by hard I mean robust ways of making decisions about whether a group of genera in a box really belong together or not. It's more robust than the linear classification system, because in the linear classification system, I can add and remove families to an order, or I can add and remove species or genera to a family, and that family still exists. In phylo you have to be very careful, because you have to be very clear which groups are in and which groups are out. If one of the groups you define to be out later has to be moved inside that box, for example, a genus being added to your box, or a whole family of genera added to your box, then your definition may no longer apply, and that will disappear. And so it makes it very, very strict in how you understand and define the evolutionary history of that group. So for that reason, it is a lot more scientific, and it is a lot less about arguments that people might have at a conference. So I've written here monophyletic clade, but understand that a clade is always monophyletic. So this is a redundancy, but it's partly for me to remind you that a clade is monophyletic. This would not be correct in a publication. You need to define the branch or stem. So this was a node definition. You can also have a branch or stem definition. This group exists if these things are in it. So if you have a bunch of genera that belong together, and you say that these genera together constitute this stem, and I'm naming this stem, is another way to do it. So you can either name the node, so it's like the box that has a bunch of genera in it, or you can name the stem that links to nodes and say everything that branches from this stem belongs to this name. This is a better way to do it if you're uncertain about the node. If this is not robust, this is probably the better way to define it. So the advantage here is that we're doing a scientific hypothesis testing, and there's clear-cut ways that your idea about the evolution of a group is either true or not true, because of the way you have to be a lot more precise about defining what is in and what is out. And it moves the emphasis towards developments in understanding phylogeny instead of the Linnean way of putting all of the emphasis on rank and authority. It avoids name changes. It separates the naming of clades from the naming of ranks. So when you add the ending that represents the rank to the name, you've linked them together, and you cannot separate them again very easily without creating a new name and a new date and authority. But here, because there is no rank part to the name, as you move it, it doesn't change anything. So going back to this paper here, and many by Cantino and the Curso explains all of this. So this is the paper where we began to discuss some of these issues in prohistology. So we're looking at node-based and stem-based definitions in phyllocode. These are how they look like. So a node-based definition is technically called a minimum clade definition. It associates with the smallest clade that contains 2 or more internal specifiers. So these are like apomorphies. The clade originating in the most recent common ancestor of A and B, or the smallest clade containing A and B, and it can be abbreviated this way when you're writing it and defining it. And then the other one, the branch or stem definition, is technically called a maximum clade definition, and it's trying to define the largest clade that contains 1 or more internal specifiers. So you'll say something like the clade that contains Homo sapiens and Neurospora crassus, it's called this. So that becomes the largest clade containing A but not Z. So you can also say if it includes Euglena gracilis, then this maximum clade definition no longer applies. So this way you have ways of using well-known groups that are well represented in the database that everybody knows are easy to find to try to define exactly what your nodes or stem definitions are. So phyllocode was published in May 2020. That was about 20 years of work that went in the background on this. There are about 21 papers published that specifically address phyllocode, but many more that discuss phyllocode. This is a rankless system without categorical names. It doesn't, you don't have to name these, but it's nonetheless hierarchical. The clades are nested, and some clades are more inclusive than others, and that's defined in Article 3. So there are apomorphy-based definitions, crown clade definition, minimum crown clade, maximum crown clade. So this is your node and stem. Total clade definition, and then you also have apomorphy modified crown clade definition. It's fairly simple. The phylonyms is all of the existing names in zoological and botanical code redefined in phyllocode terminology. So you don't have to redo it. You can just go look it up and use it or refer to this. And that's the end of this part of the presentation. This really brings us to where we're at today with phyllocode, with the UCREF database that is curated for prohistologists, and the kind of issues that we still have to discuss and come to agreements on with regards to species and populations. Those are, I think, interesting places to go over the next 10, 20 years. So if you have questions about any of this stuff, I'll address them. And then I want to move to a different topic and shift more at looking at ecology and some of the changes that are happening on the ecological front and understanding species interactions.
For now, we have not questions. We have any questions. But I was talking with some people. It's possible to make us a little break of 5 minutes.
Yes, no problem.
Okay, thank you.
So
então,
we will get together again in about 5 minutes. Okay.
Isso. Ok. Então, pessoal, 5 minutinhos de intervalo para tomar uma água, tomar um café, dar uma alongada, tá bom? E a gente volta já já. Olá pessoal. Fui falar aqui, já me engasgo bem na hora de falar com vocês. Poxa, que vergonha. Então, estamos de volta. E desculpa, sorry. Estamos de volta. E se vocês tiverem dúvidas quanto à preleção, quanto à fala do professor, vocês já podem incluir as perguntas de vocês, que aí no final também a gente vai ter outro momento de perguntas. We're back, Dr. Sina.
All right.
Welcome again.
All right, so I'm going to go through some ways we have used to approach the role of protists in the ecosystem and in communities. So this is a little bit different from what you're used to, because I work in the soil kind of environment, and it's a little bit different from working with marine or aquatic environments. But the techniques are similar. The technical problems are different. So you will see what sort of things we have been able to do to advance this. So this is the diversity. But what we're really trying to do is to go beyond describing OTUs and diversity from samples and ask some questions about why are there so many protists in every sample, and what do they do? Are they all eating bacteria? Are they all trying to eat the same bacteria, or other protists? How much are bacteria communities shaped by grazing, by protists, or by the presence of photosynthetic protists? And then numbers, for example, how much turnover of nutrients is there, and how much biomass turnover is there in the system. These are a bit more difficult to do in the soil. And there are some old techniques that had to be abandoned. We spent quite a bit of time in the late 90s, early 2000s, developing a new set of techniques that were statistically relevant. And interestingly, there are adaptations of methods that large animal ecologists and bird ecologists and some plant ecologists have been using for a long time. And these have to do with the quadrat method, transect method, and a variety of subsampling techniques that are statistically robust. And so, for example, so now we do all of these at the microscope. And so you can, your quadrat means you have known area, or you count what's in it. Transect methods, if you have fewer cells in your samples, you can follow a transect, the way bird people or large animal people do by walking in a straight line, or by flying a plane in a straight line. We just do that at the microscope and you follow it. But these are techniques that have a lot of statistics behind them, and we understand variation, and we understand calculation of mean and error for these techniques, which is much better than some of the earlier techniques that were being used. So that was important, and also making distinction between non-culture-based techniques, which were culture-based techniques were very predominant for a long time, much longer in soil ecology than they were in aquatic ecology. So we standardized some medium. I think many of you are familiar with the standard seawater solution that you can purchase the powder. Such a thing didn't exist in soil, so we came up with an artificial soil solution, which is standardized, and you can use that across experiments. And the other thing is to come up with terms that are relevant to ecology in the soil, as opposed to in aquatic systems. But we developed that quite a bit over the years, so that in the 2019 revision of the classification, the terms that are proposed there are terms that can be used across systems to describe the ecological function of the different protists. So it's no longer marine terminology or terrestrial ecology, those have been brought together, and there's a common set of terms to be used as functional groups for protists. And the ecological questions are really very fundamental. It's about who's there in terms of active and inactive species. A lot of them are encysted in the soil. They become active for a short time. And then understanding the niche space to try to figure out function in an ecological setting. So some ideas here for you to understand the dynamics of what we're looking at. And this is in the soil, but it's very similar in the in an aquatic system. There are dates here, and these are abundances. So here at these places, this is how many cells there were. On the next day, there was more. On this day, there was a little less. These are small flagellates, and these are the bacteria numbers. And as the bacteria numbers go up, these predatory consumers on bacteria, their numbers go up, these numbers go down, these numbers stay low, these numbers go up, and these numbers go up. And so you can, by looking at which species you have, you get a better sense for why they're responding slow or fast to changes in bacteria abundances. And you can trace the grazing rates from here to there. What drives all of these changes is the weather, so moisture and temperature. What happened here was a tiny bit of rain, and the bacteria numbers went up the next day and the next day. And soon after, all of these protist numbers went up because they were eating the bacteria. The protist numbers go down as they graze the bacteria, and the bacteria become dormant again. And these numbers you can do at this very crude way by looking at bacteria and small flagellate numbers, but you can also do the same thing by looking at species and species level information. Another kind of experiment you can do is by looking at mobility. In the water, currents are important, and temperature differences, oxygen gradients, nutrient gradients. In the soil, you're dealing with a labyrinth and a lot of passageways, and it's dark. Everything's by chemotaxis, and there's a little bit of water film on all the surfaces, and so movement is a lot more limited. There's no water current unless you have a very strong rain that drains things. And so we're interested to try to see how far species moved, how much they could move in a day, and what would happen if you impose a lot of water movement and mimic heavy rain to see what was the maximum amount of dispersal you could get. And so we come up with numbers like 1 centimeter per day is roughly what we get for the more mobile species. Some amoebae can move more than 1 centimeter per day. Some large ciliates will move a bit more than 1 centimeter per day. And they kind of go around and around in many different directions, but they can be about 1 centimeter away from where they started earlier in the day. That's about as far as they go on their own. So with water currents, heavy rain, and water drainage, they will obviously move much further into the streams. Dispersal, usually through animals or wind, but that only applies to the surface. The wind doesn't move anything that's below the first few millimeters of soil. And again, animal passage, people say, well, animals are moving the protists around, where they only get the surface ones, because as you go deeper into the soil, your species change, and not just the species, the kind of organisms you have change, and changes over a few centimeters. So it's very stratified and organized. They're not just randomly placed in the soil. Well, we looked at all of the primers that had been used to describe protists from environmental samples, and there's a map of it here across the ribosomal DNA range. So a lot at both ends, and then depending on whether you're looking at the V4 or V9 region or wherever, your primers will go in different places. And for some genera or families, you need to, especially for parasitic ones, you need to be very precise in being able to tell the difference between strains. And so sometimes you will find primers in strange places, but that's because it's being used for a particular parasitic genus or something of interest. This is roughly the distribution of where the primers are, and we labeled them in the same way, and we mapped them, and we looked at what happens if I do a blast against a primer from here and a primer from here across here. What kind of organisms will I detect? So this is Amebozoa, Cercozoa, Ciliates, Euglenids. This is a Silicophilosophea, and this is the Heterolobosea. And what percentage of these I detect if I use this primer set? So this was an interesting and useful paper for us to try to understand why some genera or species were very hard to get from the soil and from environmental samples. For example, these Heterolobosea, you always get very poor unless you use a particular region against another primer to get improved coverage. And these numbers are often very low in DNA analysis, even though you can see them at the microscope. And so trying to reconcile what you see at the microscope with the kind of DNA extractions that you get. Sorry. You have to do this kind of analysis. And then when you look at different samples from different places, you get, doesn't matter what the names are, it's just the colors changing. You get different proportions of organisms represented depending on whether you're near the surface or further down, or in this plot or on that plot. And so you can tell these differences down to the, as far as your OTU identification is possible. And in samples, we can tell the difference between soil profile depths, between seasons, between how the site is managed. So if there's a lot of disturbance, like agriculture, if it's undisturbed, a forest place where nobody walks, and yours in no tillage, there's an agriculture with the plow and agriculture called no tillage, which is without the plow, which is used in semi-arid climates. And this doesn't disturb the soil as much as normal agriculture. It's the reason why it's done. You can compare these sites to normal agriculture, undisturbed sites. So you can detect this level of differences, but you still don't really know what the species are doing and how they're interacting. So how do you reconstruct the ecosystem? This is an example from a boreal forest food web, northern Alberta. And it doesn't matter what the boxes are. What you have here in each box is a group of species. These are your bacteria. This is your litter, which means all of the dead twigs and leaves and the surface of the soil, the rotting organic matter. These are photosynthetic organisms, herbivores, vertebrates. These are fungal eating microarthropods. These are fungi, detritus. You just have these boxes where you have genera placed into boxes based on what they eat and how they eat. And it can get messy because this is just at a fairly crude level, but the entire ecosystem is represented there. These are grazing vertebrates on the vegetation, as is the slug beetle. And these are parasites. parasites on roots, parasites on animals. And what we can do with this, once you have this kind of structure with arrows of who eats what, you can come up with a better understanding of your system. For example, here, if I look at, oops, lost my spot. If I look at the animal parasites, there are 7 Coccidia OTUs and 12 Gregarina OTUs. And if you can identify these species, you can also know what animal is being parasitized by each one of these OTUs. And so you can get very precise about what's doing what. Here, for example, there's one centipede in the system. Haptoglossa is a parasitic organism that preys on these. And you can see that a predator like Haptoglossa in this system that is the top predator on several boxes becomes a very important species in this system because it's controlling by parasitism and predation a variety of boxes. And then you have this one centipede, which is the top predator in this soil system. The slug beetle here, this one species again becomes important. And you can identify what the important species are. Here for the filter feeders, we have 6 rotifers and 44 choanoflagellates. And I know that these and these live in the leaf litter, so it's near the surface. And the more you know about the biology of each one of these OTUs, the better you understand what exactly they're eating and what they're doing, where they're living, and when they will be active, because some of these species are fairly well known to biologists. So that was an example with a boreal forest, but we've done this in many places. Natural prairie, 140 years of continuous agriculture, 30 to 40 years of no-till agriculture, 50 years of agriculture converted to grassland, some neotropical forests. We looked at forest transects in Ecuador and Panama and Costa Rica, north and south Thailand tropical forest transects. This one is still unstudied. We have the DNA sequences, but we need to do the bioinformatics. There are 7 undisturbed forests, they're parks in Japan, and we sampled those. The work with microarthropods was done, but we still have to do the bioinformatics. A lot of things came to a standstill when the COVID pandemic came. In Canada, we were not allowed to go back to the university until less than 2 months ago. So there was 18 months where there wasn't any research. This is a site that I sampled in French Polynesia. And there's also another one which we did a 800-kilometer transect northeast China from grasslands, fairly semi-arid grasslands, to the subalpine area. And there's another one that we did in Mongolia, where we went from the north mountains adjacent to Russia to the southern mountains adjacent to China across the Gobi Desert over several days and sampled at regular intervals all the way there. That was an interesting trip because we got to camp in the Gobi Desert, and in the north we camped in some of the traditional Mongolian tents, yurts. And then this trip has been canceled twice. Once because I damaged my leg and I couldn't walk for 3 months. And then the next year there was the COVID pandemic, and so we had to cancel again. And we obtained the grant for this. We had to rewrite the grant, so that we obtained that grant just about a month ago in September. And so we're scheduled to go to the high Arctic to do some sampling for this this year. I hope this coming year. So we have a lot of biogeography covered. Most of them I think are here. There's this one I haven't marked. This one here I haven't marked. And then recent news is we're going to go up here and collect some samples And then this one is already collected. So the bioinformatics is finished for these. The bioinformatics is in progress for these. And we need to finish that before I write a big paper. Here is a recent paper that has a long history. We did this work probably about 5 or 4 years ago, and we almost got it in Nature, but then at the last minute, when we were in the second revision, Nature said they don't want it because they think it might not get enough citations. So we ended up having to, we lost about 2 years out of that process. But it's out now in Ecology and Evolution. And it modernizes our understanding of the soil food web. It standardizes the terms. It introduces some new terms. It's very similar to the one I showed you for the boreal forest. And then we have a discussion about how much diversity there is in each one of these boxes on average in a soil system. And we do a lot of simulation and testing to see how robust these interactions are and what happens when you manipulate it in this way and that way. So that is kind of nice to have out. This is the simplified way of showing it. And so those boxes here, you can simplify them into a diagram like this. And if I look at the boreal forest I was just showing you, this shows you what happens to the bacteria. The bacteria contribute to the soil solution. They're eaten by the filter feeders. These are produced bacterivores. There are some osmotrophs. Cytotrophs feed on these. So the cytoplasm in the bacteria biomass, ends up in all these different places. Predators here, the solution puts it into the plant roots. So that's how this cytoplasm ends up in the plants. And if we look at the fungi and fungivory, there's your litter, these are your fungi, these are your mycorrhizae with the roots, and the molecules here become the cytoplasm here, and they end up in all of these places here. So you can see some similarities where the predators here are accumulating molecules that they acquired from the bacteria-derived system as well as from the fungi-derived system. And both of them get their original molecules for the decomposing organic matter here. So if we look at the root consumers, these are the mycorrhizae and the plant roots. They're eaten by the root herbivores, and they're eaten again by these predators. And now you start to see that the predators here are receiving cytoplasm from here, and from the litter to bacteria through here, and the litter to fungi through here. So a lot of stuff is accumulating here. If we look at the plant herbivore pathway, so roots and endophytes and plants, they're eaten by herbivores, and they go into the predators here. And the solution here, which is from bacteria and fungi derived, gets into the plant roots and into these systems. So some of the decomposition that fungi and bacteria do becomes nutrients that the plant roots get and goes up in this direction. So now we see that we have two paths. All these end up in the predators here, and then these ones end up in the predators up here. And the osmotrophs, this is very much of a below ground system. These are obtaining nutrients that are dissolved in the water, in the soil solution. And the plant roots compete for this soil solution and the nutrients in it with all of these. So there's a lot of stable isotope work that has been done to try to figure out what goes in the plants, what stays in the soil system. And this is how we approach measuring these with stable isotopes. Instead of just tracking who eats whom based on OTUs and labeling genera and species from OTUs, it gets you this far. What you cannot do is to explain how much is going through the different boxes. And so stable isotope procedure, you take soil from here, there it is, You grow bacteria on carbon that is a rare stable isotope. So you can use carbon 13 and nitrogen 15. So the nutrients in this agar are only 13C and 15N. And you grow the bacteria on this until almost all of their carbon and nitrogen is 13C and 15N instead of 12C and 14N, which are the normal isotopes. So now you have bacteria enriched in these very rare isotopes. You inject them uniformly into this soil plug. There's your plug here. This is the injection system. And all of these bacteria end up in here. And you wait for 2 days. You can wait for a week. You can wait for 2 weeks to allow these bacteria that are now here to be eaten by the community. And you look at where this 13C and 15N has gone against the background of C12 and 14N. This is the extraction procedure for getting all of the organisms out of here. This works for the microarthropods, the microinvertebrates. You can get the nematodes out in a different way and the protists out in a different way, but you can study all of those. This is extracted protists. So what you do, sorry, not the extract, what you do is you, instead of adding bacteria, you add protists that have grown on the bacteria and you inject the protists into the soil. And this is where the carbon ends up. These are insect larvae. This is a particular root parasite. These are microarthropods, nematodes, and collembola. And so what this tells you is that most of the protist biomass, all the molecules in the cytoplasm, end up distributed this way, mostly in collembola and nematodes. These are specialists eating on microbes, so they just suck what's in the solution. These ones are eating decomposing organic matter and fungi, and they're ingesting a lot of protist biomass, but not by directly feeding on the protist, just because they happen to be in every mouthful. And these ones are also acquiring them, these mites are acquiring them also indirectly by ingesting decomposing or organic matter and eating fungi, but to a much lesser extent. And then these ones, same with the earthworms and these insect larvae, they ingest mouthfuls of soil, and in each mouthful there are some protists, and that's why they end up with this, a little bit of this carbon in it. But what this tells us is that the protist biomass, it goes to the nematodes and collembola in about equal amounts. From the collembola, some of them end up in predatory mites, mesostig mites, and this is roughly where the carbon goes in a food web from the protists. So that was interesting to find out. And then the other thing I want to talk to you about as a last thing before I make some conclusions is functional response curves and optimal foraging theory. So you may or may not know about this. If you're working with animal ecology, you will absolutely know about this. Optimal foraging theory applies to prey selection and rates of handling. It applies to protists, obviously, and I've shown a diagram here. If you can see this easily, if you spend a few hours at the microscope, there are papers that have measured it and published on this. But when you have a small protist consumer of bacteria, this is predation. You have to encounter the bacteria, you have to ingest it. If it's palatable, it will get ingested into a food vacuole and digested. If it's not, it will be ejected. So there's a frequency of encountering, tasting, and then keeping or rejecting. And this means that not every bacteria tastes the same, and not every bacteria that is encountered is eaten. There's an escape probability, and there's also a taste thing that the species might not like it. And so these are how you can break this down mathematically. So you can measure search time, and the more food there is, the less search time there is. The capture efficiency is a ratio between how many are ingested and evaded. Some bacteria are encountered, and the food vacuole begins to form, but the bacteria escapes. Just the capture efficiency. Growth rate is how much of what is ingested in the food vacuole becomes new cytoplasm, and how much of it is excreted as undigestible. And what you will find when you make these measurements is that when there's a lot of food, the food vacuole is digested very little, and things that are readily soluble are obtained from it, and most of it is thrown out. If there isn't a lot of food prey particles, they are kept and digested much longer, and a lot more nutrients are extracted from it. These affect the growth rate. It affects the duration of the cell cycle, how often your cells divide, and how fast they grow in a culture. So we can measure these things. The Pmin and Pdiv, this is the minimum prey concentration that will allow the cell to grow and divide without forming a cyst or becoming dormant. And this is the minimum prey concentration that will maintain cell division. Sometimes cells stop growing and dividing, but they don't form a cyst, and they can persist for some time in this fashion. So these are two numbers that can be identified, and they're useful in telling the difference between species and populations, because different genetic backgrounds will give you slightly different Pmin and Pdiv times. And then the other thing has to do with prey quality. If it tastes bad, it's going to be adjusted. It's not going to be eaten. And some bacteria, for example, or it could be another protist, have a lot of cell wall material. It's not easy to digest. Some of them have a lot of glycogen or oil droplets or starch, which are more useful. And so depending on what the details of your prey chemistry is, it will have a different prey quality for a different species. And these in ecology are called functional response curves. Is type 1, type 2, type 3. They're very similar to the enzyme response curves. So here I've drawn you S-shaped enzyme response curve. Functional response curve has to do with prey abundance and rate of ingestion. If you're doing biochemistry, this is your substrate concentration, and this is the rate of reaction. It's mathematically the same. You can calculate a minimum rate and a maximum rate. The cell will not feed faster than this. They can't ingest faster. You reach a plateau. And there's a minimum that if the prey is too rare, they stop feeding and they form cysts or go into dormancy. This doesn't always cross the zero. Sometimes it'll be up here, sometimes it'll be over here, depending on whether they're RNK selected. And another value that is important for niche distinction is the half rate value, half Vmax, as it's called in enzymology. Km value, which is the halfway between the Vmax and Vmin. This will vary between strains and populations and species. And you can see an example of it here. These are a variety of cercozoa. These are soil organisms that change shape. They have two cilia, this one here and this one up here. This is the same cell over that is moving across the slide. And it has some bacteria in food vacuoles inside. And we looked at 7 different strains of this. And you can see from here that they're in fact, they have to be different species. Their OTUs are different as well. one strain could eat bacteria from almost 900,000 per ml to 90 million per ml. Very big range of bacteria abundances that it can tolerate. This one here, if there are fewer than this number of bacteria, it forms a cyst. If there are more than this number, it forms a cyst. And you can see that these different strains have different bacteria abundance preferences. And so if you have bacteria abundances about here, this one will be active, this one will be active, and this one will be active. These ones will be encysted. If you take a bacteria concentration here in your sample, these ones will be active, and this one, but this one will be inactive. And so you'll see these at the microscope, but not this one. These become numbers that are hard to obtain, but once you have them, tell you a lot about how these species are different. And so these are the numbers the values I was showing you before. Vmax, Vmin is the Vmax, Vmin in the previous slide. Cell cycle duration, 2 hours to 6 hours. But when they're not dividing but feeding, you get these long cell cycles, 11 to 15, and eventually they might divide. So it slows down quite a bit. Some of these species have fast cell cycles in the 2-hour range, especially at the higher bacteria abundances, but some of them are slower dividing, and every now and then you have a species that might be very slow dividing. Range of ingestion, this was bacteria per minute per cell. At Vmin, it's 0.2, and at Vmax is between 3 and 4 for these different species. And it really has to do with the size and volume of the cell. It's hard to put 3 or 4 intact bacteria into these cells without causing a lot of disturbance. There's just no room. And the fastest you can make them grow, eat, is 10 to the 8 bacteria per gram. Just more than 10 to the 8 bacteria per gram is what they can handle. That's where they saturate. So you can calculate daily consumption rates. 3 times 10 to the 5 bacteria per day per gram of soil. And this would be by one organism. And at the maximum rate, you can get a number of 6 times 10 to the 11 bacteria per day per gram of soil. And what these numbers tell you is that basically all of the bacteria in the soil are eaten about once a day. And they have to regenerate at that pace in order to keep up with the grazing rate. When there are fewer bacteria, there are fewer grazers on it. That's similar to this one slide I showed you at the very beginning. And when there are more, you get a lot more of these, but then their numbers go down again. And you can do the same with amoebae. There's a nice vanilla there. We looked at 8 species from common genera, but from very different locations. And what you realize is that some of the numbers are similar to those Cercozoa, but some of them are not. Bacterial selectivity varies more between species than they did with Cercozoa. Ingestion rates were more variable. But overall, they're less sensitive to bacteria abundances. Minimum consumption rate was, again, about 0.2 bacteria per minute, just under. Maximum consumption rates were much higher, more than 15 bacteria per minute for these very small amoebae. And it's interesting how they taste and choose what they want. They will crawl on the agar plate on a bacteria colony, and if they don't like the taste, they will just keep moving and the bacteria colony stays there. But if they do like them, they will stay there, and when they move on, that bacteria colony is gone. It will all have been ingested into food vacuoles. And because of the way they eat, they can make food vacuoles at several places and ingest a lot more bacteria per minute. So that was those were interesting numbers to obtain. Their cell cycle duration is more than 10 hours, and it can extend to about 36 hours for low prey. So we don't get fast divisions with the soil amoebae the way we do with some of the cercozoa or ciliates. This one was looking at ciliates. So this is the same cell, and it is feeding here on Chlamydomonas. They go through the oral structures and accumulate as food vacuoles here. The color changes have to do with how decomposed they are. And then the undigested part gets excreted through here. This is the same cell growing on bacteria. It becomes much smaller, and you can see the bacteria inside. These were stained. And it adjusts its size depending on what you're feeding it and the concentration of what you feed it. And so we did an experiment where we changed the temperature, 4 degrees, 17 degrees, and 27 degrees, and we offered different prey particles. KP is a bacteria. This is a small heterotrophic nanoflagellate. This is Chlamydomonas reinhardti, and this is a small ciliate Colpoda steini. So this cell can ingest a colpodid, which is about a quarter of its size, but at a very slow rate. It can ingest bacteria. And when you look at the rates and biomass of this predator on consuming prey, you end up with a chart like this. So at 27 degrees, this is the biomass growth that you get on Bodonid, on this heterotrophic nanoflagellate. And Chlamydomonas reinhardti, 17 degrees, gives you the most, the highest rate of growth. Next is this one, the next is this one and this one. And it never grows very well on Colpoda steini, which is a little bit too big for it. The other thing this tells you is that not only is it adjusting its size to the prey and temperature, but also to the abundance of these. And when it's at 17 degrees, in the presence of all these organisms, these 4 food choices, it will preferably eat Chlamydomonas and fairly fast. But at 17 degrees, at 27 degrees, which are these ones, it prefers to eat the heterotrophic nanoflagellate. At that temperature, Chlamydomonas is too fast for it, but this one is not, and so you prefer to ingest this. And at 4 degrees, the preferred organism is again Chlamydomonas reinhardti, because it becomes very slow and not escaping the ciliate. So these kind of numbers are useful when you're looking at one species, but they also help you distinguish between morphologically very similar species that are, they look like they should be doing the same thing, but when you actually map this out, you will see that they are not eating the same way, and they have different organism size preferences for prey and different temperature preferences, and that we know is true. So finishing up here, these functional response curves, we have very few of them, but they're very important for making models and understanding rates of nutrient turnover in the system. And these kind of numbers at the microscope can be verified by stable isotope studies, which are less precise in some way, but tell you more in another way, in that they tell you where exactly the nutrients are going in the community and system. Proteins are ignored in more solid food web studies because they're hard to work with in the soil. You can't just put it at the microscope. You have to extract them first, but it's possible. So I'm going to finish here with about 5 slides, asking the question, what shapes the diversity of species? These are mites and colembula in the middle, microarthropods. Are most species rare, local species, or cosmopolitan? This was a debate at the beginning of the 20th century. couple of people tried to raise this discussion again, but we have a lot of biogeography information now from species sampling around the world, and we have a better understanding of that. Are trophic functional groups determined at the genus level or family level? Again, this is something we can begin to answer. With some protist groups, it's at the genus level. In others, it's at the family level. Both of these are correct. And how to reconcile classical field sampling designs for species richness analysis by DNA sequences, because the field sampling design usually requires a lot more volume and bulk collected. But for DNA analysis, you collect very, very little sample, very small amounts, about 0.5 gram or 1/3 of a gram, very tiny amounts. And so statistically, the robustness of these two techniques are different. And the DNA sequence analysis is more expensive, but if you sample well, you get a lot of information. The microscopy procedure is very cheap, but very time consuming, and nobody has really enough time to do it this way. And so you have to find a balance between how much analysis you're going to do and how much depth you're going to get in your analysis. And a lot of the genomic studies and DNA extraction studies suffer from too much pooling of samples and poor subsampling of the DNA. You lose heterogeneity across your sampling site as a result of this. So 3 rules. Most eukaryote species are rare and local. One example here, that's from that paper. I think this is something that people will agree these days. It's the first rule. There's a lot of functional redundancy. And so if you go back to those boxes that I showed you, there are a lot of species, a lot of genera in each one of those boxes. And they're eating the same thing. So there's functional redundancy. But why is there functional redundancy? It has to do with functional response curve changes, rates of growth, rates of ingestion, and preferences for month and season and temperature, things like this. And that's where cryptic species become important. The morphology hides a lot of cryptic species. They look the same, they seem to eat the same thing, but they're not the same strain or species or population. They do different things. And there are many papers that have now accumulated arguing that. Second rule that we can take away from this kind of information, this is a small terrestrial flatworm. It ingests organic matter. Not all species are active at the same time, and that's true in any system. But through the year, everybody gets to be active once in a while. That's why in your samples, you have some species that are active and they seem to be absent at the microscope, or very rare in your DNA sample. And then you go there a few months later or a few weeks later when the weather is a bit different, and you'll find a different species composition and proportions. So not all species are active at the same time, but everyone gets to be active once in a while. One implication of this is rare species. Rare species are rare and encountered infrequently because they get to be active rarely. They still belong to the system, but the conditions for them to be active occur rarely. Now, with climate change, all this shifts. You will get community structure changes as the weather changes through the year and so on. So species composition varies with time and place. And then the third rule, the last one, and that is particularly true for the soil, where you have a lot of structure as you go down or across. You go from a place where there's a leaf from this tree and then a leaf from that tree. Then you have a dead fungus, and then you might have a dead insect. So in all directions, you have heterogeneity, spatial heterogeneity. A PED is a soil aggregate. And these determine the habitat. I think this is the last lesson that people are coming to grasp with. So here I've given you again that vanilla, and this is a pseudoscorpion. They're about 1 or 2 millimeters long. They are scorpions. They're missing the tail, so they're not poisonous. They're fairly easy to find. You'll have a few per meter squared if you're in a forested region. And they feed on small microinvertebrates. They're predatory. Very interesting biology. So what structures these environments and determines who is present and what will be active really has to be looked at the millimeter and sub-millimeter level. And I think that's where a lot of our conclusions are going. And the recovery of the soil structure is the rate limiting step in site recovery. So if you have an agricultural field or a mine site and you want to go back to a forest, the time it takes to develop the heterogeneity in the soil structure is the rate limiting step. And the microhabitats become determined in this aggregate. And it drives species establishment. When the heterogeneity returns, the diversity of niches can be accommodated, but that can take 10, 20 years. And so I'm going to finish with this. This is an aggregate of soil, but it could also be a detritus particle in the ocean. There's an oxygen gradient. In the middle, it's anaerobic. So you will have anaerobic bacteria. Here you have microaerophilic environment. So you'll have different bacterial metabolism. On the outside, you'll have lower oxygen, and then 21 is the amount of oxygen in the atmosphere. And so on the surface of an aggregate, you have aerobic respiration. And this particle is about 1 centimeter in diameter. So this is what we're looking at when we're in the soil. This is a marine coastal sand stratified. You can see the different colors, 2 centimeters. And each color represents a different bacteria with a different metabolism. So you get this structure horizontally and vertically in a coastal system. This is a soil aggregate that was measured with microelectrodes. And this structure that you see here, this stratification of bacterial metabolisms is exactly what you get here as you go from the surface to the inside. We encounter this again in marine sediments. And I've shown this. This is a scanning micrograph of a plug of soil that shows you the aggregates. There are some pebbles and there are some aggregates. And each one of these aggregates has this distribution of oxygen, and therefore bacteria will be organized from the center to the outside of one of these particles in this way, as we have in marine coastal sands, for example. So this is how the environment becomes stratified with different kinds of bacteria and different protists that ingest those bacteria, and the environment they have to accommodate is at that scale in the soil. You get similar system in the marine environment, but the scale is bigger so that your oxygen gradients are over tens of meters. So these are, if you wanted to do ecology and move away from phylogenies, a lot of that work is now finished. These are the kinds of things that can be done to try to understand the role of different species and of different kinds of organisms in protist communities. All right, I'll stop with this. This is a journal that I'm the editor of. We started this journal in 2016 to focus on soil, organism, and root nutrient uptake kind of interactions. I'll end there. So if you have any questions about the ecological part, let me know.
Okay, we have some questions actually about this last part and about the other part. Professora Ina ask, how long does it take to build a tree?
A phylogenetic tree?
Yeah, phylogenetic tree.
It depends on how many sequences you're comparing and analyzing, and the length of your sequences, what the depth of analysis sorry, I'm getting tired, of DNA sequence analysis was, and the kind of computer you have access to. With some of the clusters, if we something that will take a week on a laptop, you can do in a cluster in an hour or two. So it really depends on how much information you have and you're comparing what with what.
Okay. If you prefer, you can go out of the presentation and come back for the window of the StreamYard.
All right.
All right. Another question. Some years ago, one of the main limitations for the construction of phylogenetic trees was computers and hardware. Now, this seems not to be the major of problems. How are the challenge now for big and robust phylogenies today?
You need access to a cluster, and I think most universities will have free access to, as part of the university infrastructure. There are companies that will do that for you. I think the limiting factor now really is having good bioinformaticians that understand the biology sufficiently to know what they're doing. There's a lot of demand for bioinformaticians, and I've had postdocs who were very good and postdocs who were not so good at not so much the bioinformatics, but the interpretation and knowing if they're on the right track. I can work with, I remember one postdoc spent 2 years generating junk. And then when we repeated the work with another postdoc, we managed to do everything in a couple of months and verify things and do it properly. I think it really depends on the bioinformatician and what kind of background they have. So you're right that I think neither the sequencing or the computer is really the problem anymore. I think most of it has to do with getting good bioinformaticians that will work well with biologists.
Right. Did you say that, well, we know also that the soil have a complex community and complex relationships. Yesterday, we had a lecture about possible evidence of life in Mars. And, well, do you think that it is possible to reproduce this relationship and construct in a future a true soil in extremophile place like Mars, for example?
Yeah, I think one difference will be availability of organic matter for digestion as a source of carbon and nitrogen and other elements. If you want to have carbon-based life, you're going to have a source of carbon. So if you assume that there's no active life now, but there was a long time ago, I don't know if there's enough DNA or microfossil traces to find. If you think there's some today, then there has to be some kind of source of carbon and nitrogen for them. And I don't know what that is.
Maybe cyanobacteria in the future.
Yeah, the bacteria have to obtain there'll have to be some form of autotrophy to generate biomass. And maybe you only have interacting bacteria without predators, but you should be able to find colonies here and there if that's the case.
Because we saw a movie that a man, I don't remember the name of the actor, plant potatoes in Mars, but that is not possible today because the soil is extremely poor there.
Yeah.
Actually.
Yeah.
Great.
You will have to be almost hydroponic water.
Oh, we have another question here. Felipe Benitez again. Can you see the question?
Yes.
On the screen?
Yeah, no, I think this is very important to do. Yeah, there are a lot of unculturable genomes. Yeah, no, I think the loci have been very, very important for understanding early eukaryote evolution. And there are many eukaryote genomes that we know from DNA, environmental DNA, but we don't know what they look like or how what they do. I think that's part of what we're doing these days is studying a lot of these, finding a lot of these environmental DNA, and then we spend a lot of time trying to find the organism.
Great. We have another question, people. Well, I would like to thank you so much, Doctor, for your presentation, your lecture. We have a lot of data, and it was interesting because some days ago we have some questions about phylo that you brought for us today and explained something about that doubt about phylocode that, well, it's now evident in the present that we have difference in the classification, in the classifications. Then I would like to thank you so much, and I would give a short time for last words for us.
I think the last 30 years, most protistologists were trained to do phylogenies. A lot less electron microscopy. I think a lot of that work is done. There's still a lot of very interesting phylogeny work to do, but the bulk of the questions have been addressed. Where things are going to get interesting for the next 20, 30 years is doing a lot of comparative cell biology. There are genes that make proteins that are very rare in humans and animals, but they're abundant in protists. For example, some of the cytoskeletal elements, some of them implicated in some cancers, are very rare in a mammalian cell, but some of those will have their origins in protists, and in some protists they will be abundant. For example, the excavates have a lot of cytoskeleton. Around 99, 2000, I had a small grant and a postdoc to look at some of the oncogenes in protists, and we could find them. And so we know that those genes have their origins traceable in protist lineages. But at the time, the cell biology instrumentation and technology or molecular biology wasn't advanced enough for us to do anything with it. Today, you can do a lot of really cool cell biology and genomic work with single cells. And it is now possible to go back and ask those questions again and have a look at genes in a comparative cell biology way that are abundant or easy to work with in some protists that will have implications for how we understand them in cancer and mammalian cell biology. So I think that's one aspect of protistology that we'll see grow. And the other one has to do with understanding nutrient transfer in communities across ecosystems. We have a lot of people willing to put a lot of money in preserving biodiversity. We have a lot fewer people able to describe why that biodiversity needs to be preserved. We kind of moan about the loss without having solid arguments for politicians about why they should preserve it. And the more we understand why we need many species in each functional box, the better we'll understand what provides community stability and ecosystem function sustainably. So some of that microscopy and stable isotope work there's a lot that can be done there in trying to understand the planet's biogeochemistry and mitigation of climate change and remediation of and impacted environments by pollution.
Great.
Yeah. So I would encourage, I think I would encourage new protistologists to think a little beyond the phylogenies and more into cell biology and ecology.
Great. Thank you so much for your words. And I think that that is very important for us to stimulate the research about protists, about algae here in Brazil, and also connecting with the research of other research around the world. Thank you so much.
Sure. Maybe I can say one last thing. I didn't mention this, but I said that there wasn't very much research going on during COVID because our universities in Canada were closed for 18 months. One of the side effects of that is that I have now almost finished writing a protistology textbook. And the textbook will be finished and with the publisher November, December. It's pretty much finished.
Great.
I'm working with some illustrators to work on some figures, collecting some photos, finishing some parts of the text, but it's essentially finished. And it should be available by June. And if there is anybody who's interested in spending some time translating it, I have someone who is translating it into French, another person translating into Japanese. But if there's someone who wants to translate it into Spanish or Portuguese, that would be something we can start working on almost right away. It'd be a bit of time, but you'll have your name on a book.
Oh, great. We'll talk about that. It's very important because one of our discussions during this course was the lack of sometimes bibliography and textbooks about some issues, and especially bibliographies for undergraduation students.
Yeah.
And it is very important for the formation of new people, of new biologists and new protologists.
Yeah.
I think that is very, very important and interesting. Thank you so much again.
Thanks so much.
Ok, pessoal. Então.
Thanks for the invitation.
Até mais. See you later. Pessoal, então, agora nós encerramos as nossas participações. Nós tivemos, então, uma série de 19 apresentações até hoje, no final do curso, desde o nosso início. Uma apresentação, infelizmente, nós não pudemos completar, que foi a da Priscila, em razão de problemas médicos, né? Outro resumo, outra síntese do nosso evento é de que uma das programações também que foi feita anteriormente, nós não conseguimos fechar, que foi a do professor Robert Anderson, por conta de problemas de saúde da esposa. Então, são essas as duas justificativas, mas em geral nós tivemos, então, a participação de todos aqueles que se propuseram, né, vir e participar conosco, estar conosco. Esse ano eu contei com a ajuda de 3 pessoas muito especiais, meus alunos, a Vanessa, o Tiago e o Zé Otávio, que estiveram aqui comigo, auxiliando, então, nesse manejo aqui, que às vezes é um pouco complicado, porque o negócio aqui é assim, às vezes trava e eu tô clicando aqui, não vai, aí já vai a Vanessa e coloca, né, ou vai algum dos meninos que faz uma preparação. Foi uma preparação que a gente começou já desde o meio do ano, então fazer as publicações, né, fazer os anúncios, fazer os convites. O ano passado a nossa participação foi maior, sincronamente, e até mesmo eu estava conversando com alguns colegas a respeito disso. Muito disso se justifica porque o ano passado ainda muita gente estava muito ainda perdido diante da pandemia, das atividades que estavam sendo realizadas, ainda havia poucos cursos online, e esse ano, né, ou nessa transição daquele final de ano para cá, nós já tivemos um aumento estrondoso do número de cursos, do número de atividades online, que se tornou extremamente cansativo, exaustivo, né, para muitos aqui, uma série de reuniões. Então, a nossa participação online, ela foi maior nos primeiros dias e foi decrescendo nos últimos dias. Ainda nós temos a problemática, talvez, da língua estrangeira do nosso curso, e aqui eu justifico, meu inglês é terrível, mas mesmo assim eu tento conversar, eu tento aprender, eu tento falar, entender um pouco, e eu acho que a gente tem que estimular. A gente tem que estimular esse tipo de participação, porque se a gente for esperar, a gente está perfeitinho para conseguir falar, a gente não vai conseguir. Além do que, eles falam inglês, muitos deles falam inglês perfeitamente, alguns falam inglês, talvez com determinados erros, mas quando vão falar português também erram um monte. Então, quando vão falar espanhol também erram um monte. Então, estamos no mesmo patamar, né? E isso é extremamente... Mas o que eu estou justificando isso é justamente para a gente estimular e tentar pensar nessa questão da língua estrangeira. A gente tem que se linkar, juntar com esses grupos, com esse nível de pesquisa, para a gente conseguir, então, incrementar ainda mais as nossas pesquisas aqui no Brasil, talvez ganhar suporte, né, auxílio, fazer parcerias. Está aí uma parceria que o Sina propôs aí já para nós, para quem estiver interessado, fazer a tradução do livro do Textbook of Portuguese que eles estão preparando, né? Então, depois eu vou aproveitar os e-mails para vocês, e os e-mails de vocês, e vou mandar um formulário de avaliação, né? Que eu acho que seria interessante a gente pensar o que foi positivo, o que pode melhorar, as falhas que nós tivemos, pedimos desculpas, naturalmente. Vou trazer a Vanessa para cá, ó. Vem para cá, Vanessa. Vanessinha, que ajudou muito, o Zé Otávio, que não está aqui, mas o Tiago também, que não pôde estar, cada um com sua dificuldade, né? O Tiago com problema no notebook dele, que não tinha câmera, não tinha microfone, então às vezes entrava, não conseguia fazer nada, mas fazia os posts lá do Lamic, né? Então, agradeço imensamente a vocês e agradeço a eles por essa participação. E se a gente conseguir esforçar bastante, no ano que vem eu vou estar com uma série de outras atividades, né, maiores ainda, mais pesadas, mas eu espero que dê tudo certo para que talvez a gente consiga organizar um terceiro. E nesse terceiro a gente melhorar ainda mais. E quem quiser até mesmo auxiliar na organização, participar, não, vamos fazer junto, vamos, né, tentar incrementar, melhorar nisso, melhorar naquilo outro. Vamos pegar junto e vamos fazer para a gente conseguir, sei lá, incrementar mais o nosso conhecimento. Para mim, mais do que, não sei para vocês, assim, né? Mas para mim, que estou mesmo aqui do outro lado, gente, aprendo pra caramba. Então, assim, estou aqui de camarote, né, aprendendo com vocês. Aprendendo com Vera, aprendendo com Mariângela, com o Carlos Bicudo, aprendendo com os internacionais, aprendendo com Orlando, com Ina, né? Então, é um prazerzaço estar aqui com vocês. E aí, então, fiquem de olho, nos próximos dias nós mandaremos as avaliações para vocês e logo depois também nós enviaremos o link para vocês retirarem os certificados de vocês. Para a retirada do certificado, para quem já fez, né, a inscrição, eu já anuncio que vai precisar simplesmente do número de inscrição e CPF de vocês. Aí vocês colocam no sistema, já vai aparecer o certificado, vocês podem fazer o download ou em preto e branco, né, para quem já tem talvez que anexar no currículo Lattes, impresso e tal, ou colorido, tá bom? Então, agradeço imensamente. Quer falar alguma coisa, Vanessa?
Só que foi engrandecedor, sim, participar, conhecer gente nova e dessa vez participar ativamente, né? Porque o ano passado eu estava como aluna e aí esse ano a gente aprendeu, além das palestras que foram diferentes, esse manejo e conversando com pesquisadores que antes eu não teria contato se não fosse pelo backstage.
A Gisele aqui perguntou se no ano que vem a gente não poderia incluir a marinha, algas marinhas. Bom, o curso nasceu, Gisele, inicialmente vinculado a uma disciplina que eu ministro na UEL, que é de comunidades de algas continentais, que é justamente de comunidades de algas de ambientes terrestres, né? De ambientes interiores, vamos dizer assim, não de ambientes terrestres, ambientes interiores. Mas, de repente, para o próximo ano, a gente pode vincular também. Então, a gente pode ir conversando, verificar. Tem muita coisa legal para a alga marinha que a gente pode trazer também. Né? Até mesmo fazendo link aí com esses grupos de aguador. Se vocês tiverem mais sugestões, mais dúvidas, também podem mandar para o e-mail lá do LAMIC, tá bom? E eu encerro por aqui, deixando vocês descansando depois de 2 semanas, né? Depois de 2 semanas que foram legais, mas foram cansativas. Eu espero que todos fiquem bem, que continuem tomando cuidado, continuem usando máscara, continuem se protegendo as suas pessoas queridas, tá bom? Tomem vacina, por favor. A taxa de eficácia está muito boa. Então, tomem vacina e a gente vai vencer isso. Vai vencer isso. Então, até lá. Até um abraço. Fiquem bem, pessoal.